Okay, so welcome to lecture 26. Just another quick note, I'll put this up a few more times. Don't forget that our exam is face-to-face, and it's taking place on Monday, December the 20th, the last day of exams, from 2 to 4 in McKinnon Gym, and that's for all sections, even if you're in the online section. I know there's about 20 people in the A03. It's also taking place at the same time. The exam will be, in this case, all multiple choice because I have to have the grades in by, I guess, that Friday or something. So it wouldn't give me time to get through 500 of them before that. So it'll be very similar. Again, I'll tell you more information about the exam either late next week or early the following week. I'll let you know about how we're going to work this sort of the cheat sheet that you can bring in as a sort of a memory guide, and I'll take you through the process for the bubble sheets and the process for handing in and all of that, because I think for many of you it will maybe be your first, well, this term will be the first time that you've done the face-to-face exams. But it won't be your first exam because it's the last one at the university, I think, this year. So, luck of the draw. We talked about G-protein coupled receptors, and G-protein coupled receptors are an extremely important family of receptors. They all share the same general structure, which is there's 7 transmembrane regions. This means that there are a series of loops, and importantly, these cytosolic loops here, the ones in the cytosol, these are important for acting as the GEF for the heterotrimeric G protein. This slide's also indicating some of the possible types of post-translational modifications we might see that can modify the activity. And we know that it's important to be able to modify the activity of these receptors because we're always trying to reset our baseline. Okay. So the actual structure we can see here, and all of them look like this. There's these 7 transmembrane alpha helices, and their positioning with respect to each other is important. Upon ligand binding, or in the case of the eye, a photon of light coming in, what happens is that the relationship between those helices changes, and that causes changes in the cytosolic loops, and that allows them to act as the equivalent of a GEF. It allows them to kick GDP out of the alpha subunit of the heterotrimeric G protein. And so we did the comparison of the monomeric and the heterotrimeric GTPases. Okay? And again, there's lots of similarity. For the heterotrimeric, the gaps, so the catalysts that speed up turning them off, they are replaced by enzymes called regulators of G protein signaling, or RGSs, and the GEF is replaced with the G protein coupled receptor itself. Bless you. So the process of activation is shown here, and what we have is this heterotrimer, an alpha, and then a beta-gamma subunit that are associated together. The gamma and alpha are lipidated so they can remain associated with the membrane. Upon ligand binding, there's that conformational change, and the GDP is kicked out, allowing GTP in to bind. The GTP bound alpha subunit can influence downstream pathways, and the alpha beta can as well, although we didn't look at any specific examples of that one. In this example, the alpha subunit is mediating the activity, regulating the activity of whatever this purple blob is. The alpha subunit has intrinsic GTPase activity that will hydrolyze GTP to GDP. Again, this can be sped up by an RGS. And this alpha GDP bound form is now in the off state, and it rebinds beta-gamma subunits. We looked at a number of different alpha subunits, but really the one we focus on is G-alpha S, which is used to turn on adenylyl cyclase. Today we're actually going to look at this one here, G-alpha Q as well. But importantly, there are 27 different alphas, 5 different betas, and 13 gammas, and these can be put together different ways. And we call this combinatorialism. So they can be put together in different combinations. This allows us to use a smaller number of genes, but to have a high sort of level of organization or combinations, I guess, so that with a small number of genes, we can get more possible outputs. And that's a theme we keep kind of coming back to in our signaling section. We then looked at the fight or flight pathway. So the fight or flight pathway involves epinephrine as the signal and beta-adrenergic receptor, and this is used by muscles to free up glucose from glycogen. Remember that glycogen is the storage molecule that we use for glucose in the cells. Actually, the pathway is the same as glucagon, which is the signal, and the glucagon receptor in the liver, where what happens is glucagon is a signal that indicates there's low glucose in the blood, and it causes the same thing, freeing up of glucose from the glycogen in the liver to occur. Remember, though, that the liver can actually export glucose, and so it, unlike a muscle cell, is capable of pushing the glucose out into the bloodstream, thereby balancing the blood glucose levels. Yes?
Yeah, I was going to say, like, when the fight or flight response is triggered and you release all the glucose, once it like calms down, would your body restore the unused glucose into glycogen?
It depends how much is left over. So the question is, will the glucose go back into glycogen? And it depends how much is used, because if there's excess glucose in the blood, what's going to happen is, in fact, that will trigger insulin pathway, which is the pathway we're going to look at a little bit later today. This ultimately results in the production of a secondary messenger called cyclic AMP. I shouldn't say ultimately, because this then goes on to activate protein kinase A. And many of the effects we see are the result of protein kinase A activation. So protein kinase A is an extremely important enzyme for regulating glucose metabolism. We look at it a bit here, but we spend a lot of time on this in, say, Biochem 299 or Biochem 300b. One of the ones that's a little bit tricky sometimes for students is this concept here. So again, if you're looking at phosphorylation, that's a very fast sort of reaction that can occur, and that phosphorylation may turn something on or something off. But protein kinase A can also regulate gene expression. And so originally there was a number of genes identified that were sensitive to cyclic AMP levels. And when analysis was done, it was found that they all shared a sequence in the promoter region. And so this was called the cyclic AMP response element. It turns out that this doesn't bind directly to cyclic AMP at all. It's an indirect effect. The cyclic AMP response element is bound by the cyclic AMP response element binding protein, which we call CREB. CREB also, at this point, isn't doing anything significant. CREB gets phosphorylated, and now it's bound by the CREB binding protein, which is the cyclic AMP response element binding protein. And this serves to recruit RNA polymerase, and that initiates transcription. So this turns the pathway on. Phosphorylation of CREB activates the pathway via CBP. Any questions from last class? Okay, today we are going to look at phospholipase C and calcium signaling, receptor tyrosine kinases, and lipid signaling. This is where we got to this morning. Again, just a last reminder, we do have a social hour tonight. If you're interested, via Zoom at 6 with James and Elisa. They're going to tell us. I just called it an evening of fertility because they didn't give me a They didn't give me a title, so I'm to blame for the somewhat dull title, but it will be an interesting talk, I believe. So if you're interested in medicine or working in a medical laboratory and you want to know how to sort of pursue that, or you're interested in fertility treatments, this will be one for you. So here we have phosphatidylinositol. Okay, so this is a phospholipid. I gave it away. I was going to ask you if it was a phospholipid because it's got a phosphoryl group here. Is it based on glycerol or sphingosine backbone? Who says glycerol? One person? That's it? Sphingosine? No people? Well, the class was overwhelmingly correct in that it's a glycerol backbone. Okay? There's a phosphoryl group or a phosphate, and we know that that interacts with the hydroxyl group of the head group, and in this case, the head group is something called inositol. I've corrected the structure here a little bit. The lines were, for some reason, pointing a bit funny like this in the figure in the notes. I'm not sure why, so I'm not going to ask you for that, but just something that you may want to correct. Inositol, this is carbon 1, 2, 3, 4, 5. It can be phosphorylated on positions 3, 4, or 5. And this particular example is phosphatidylinositol 4,5-bisphosphate, or we can just call that PIP2. Because it's phosphorylated at positions 4 and 5, and we remember if something looked like this, that would be a diphosphate, but this is a bisphosphate because it has 2 individual phosphoryl groups. The way phospholipase C works is it cleaves along the dotted line here, and this leaves diacylglycerol behind in the membrane. So this line here is indicating the membrane. This is the membrane. And so diacylglycerol is left behind in the membrane. We remember talking about that when we looked at our lipid section. And the phosphate group goes with the inositol. So this is now what we call IP3, or inositol 1,4,5-tris phosphate. There's 2 ways that we can activate phospholipase C. This is showing activation via a GPCR. So this is a GPCR here. And what happens is we activate what's called G-alpha Q, and G-alpha Q activates the phospholipase. There's different isoforms of phospholipase C. Isoforms come from events where a gene duplicates. They maintain their function, so all of these isoforms act as a phospholipase and cleave in the same place, but they have slightly different regulation. And in this case, phospholipase C beta is activated by a G-alpha Q. Okay? It's residing in the membrane, so it's easy for it to start chewing and clipping off those IP3s. In contrast, via what we call receptor tyrosine kinase signaling, that activates phospholipase C gamma, which does exactly the same thing. Okay, so there's no difference once it's activated. So the 2 isoforms function the same way, but are activated by different pathways. IP3 can open up calcium channels on the smooth endoplasmic reticulum. So this is showing calcium regulation. In the cytosol, the concentration is about 10 to the minus 7 of calcium. It's much higher in both the smooth ER and the extracellular space. It's about 10 to the minus 3. So IP3, what it does is it opens up these calcium channels and calcium floods into the cell. A very fast reaction. I should point out that that little diacylglycerol component in the membrane also is a signaling molecule. but we're not going to discuss what it does today. You'll look at that in Biology 360. So there's actually two signaling molecules produced here, one that's cytosolic and one that remains in the membrane. Calcium levels are maintained by a number of systems. So here we're using hydrolysis of ATP to pump calcium either out of the cell or into the smooth ER. Is that direct or indirect active transport? Who wants to guess? Remember that's, yeah? It's direct, that's correct, because we're linking it to a chemical reaction. In contrast, the sodium exchanger, about 3 sodiums go in, I believe it's 3, and 1 calcium goes out. So that's indirect. And this is an example of an antiporter, where sodium is going in a favorable direction, but it's pushing calcium in an unfavorable direction. Okay? So here's an example of a very fast reaction that we see with calcium signaling. So this is looking at egg fertilization just in time for tonight's talk. And so one of the things that happens is we want a single sperm to fertilize an egg. And one of the mechanisms for maintaining that is shown here. And so after fertilization, kind of a jelly-like coating forms around the fertilized egg, and this is called the fertilization envelope. And this is caused by release of the contents of what are called cortical granules. So this is an example of a signal-directed exocytotic event. So these cortical granules are sitting just under the plasma membrane of the egg. Okay. Here, this is where the sperm comes in, and this is looking at a dye called aquarin. And this fluoresces in the presence of calcium. So we can see within a minute, we have calcium kind of at the point of contact. This then sweeps its way across the cell, and in just a little over 2 minutes, this calcium wave has spread through the cell. The cortical granule contents have been released, and we've established a fertilization envelope around the egg, which prevents entrance of other sperm. This is a very transient effect we're looking at here. To extend the effect, make a longer effect for calcium, we can utilize a protein called calmodulin. Calmodulin is a highly conserved protein. What this means is if you compare a large number of organisms that have calmodulin, the sequence is very similar between all of them. That's what conservation means. That often indicates that something has very important function. Because if an organism, say, had some sort of mutation or perturbation to that gene, and it no longer exists, it means that it decreased their fitness. Okay? Calmodulin itself, and this is important, and I often ask this on exams, of which we have one coming up, it doesn't have enzymatic activity. Instead, it's a calcium modulating enzyme. And in the presence of calcium, it modulates the activity of enzymes. It can take up, I mean, there's a lot of this, up to 1% of a cell's total protein mass. Okay? And it has multiple configurations. The first is the unbound form. And then it binds to 2 calciums. And once it binds to calcium, it shifts into a conformation that allows it to bind to its target. Once it binds to its target, it shifts shape again, and now it modulates the effect of the target. There's a number of different processes that are mediated by calmodulin, okay, including regulating the vision pathways. There's an important role for calmodulin. Another is that they can moderate the effect of CAM kinases. There's a number of different CAM kinases. This stands for calmodulin modulated kinase. The one I'm going to talk a little bit about is one called CAM kinase 2. So CAM kinase 2 is only active in the presence of active calmodulin. So thereby it means this is only turned on when there's calcium has been present. CAM kinase 2 is quite interesting because it's been implicated in the formation of memory. And they've done animal studies where they expose an animal to an event, some sort of event. If they then inhibit CAM kinase 2, the animal forgets that event. And so there's been some research being done on this as a possible drug to give to people following some extremely traumatic event, so the person loses the memory of that event. But it's also really interesting from the point of view of understanding how memory might form. So now we're going to move on to protein kinase receptors. And remember, we have two types of these. In some, the receptor itself has kinase activity, and in others, the receptor is associated with a protein kinase. From the point of view of what it looks like, it's basically the same all the way along. It's just where the kinase activity resides. So for these, the cytoplasmic domain has kinase activity. There's a transmembrane region, and then there's an extracytosolic or extracellular receptor component. They're usually single-pass transmembrane proteins, so they just go through once, unlike the GPCRs, which just keep going through millions and billions of times, or 7. Upon activation, they multimerize. So commonly, what we're going to look at is going from a monomer to dimer, and then for TGF-beta signaling in Friday's class, we'll look at them going from 2 individual dimers to a tetramer. The classes are tyrosine kinase receptors and serine threonine receptor kinases. Okay? And this brings us to a kind of a really interesting and important point. So first of all, not all protein kinases are receptors. Okay? So we need to delineate or make that difference. There's lots of cytosolic protein kinases, for example. But if we look at the difference between these, we talked about this idea of complexity and how multicellular organisms are a lot more complex and have more complex signaling systems than unicellular organisms or microbes, things that exist as a single cell. If we look at multicellular organisms, they have lots of tyrosine kinases in the genome, both receptors and non-receptors. However, if we look at things like yeast that are unicellular, there's very few tyrosine kinases have been identified, interestingly enough. These protein kinase receptors are often bound by what are called growth factors. And growth factors are important for growth and division, okay? And the Nobel Prize, there's been a few, but the early Nobel Prize for identification of these went to Stan Cohen and Rita Levi-Montalcini in 1986. Okay? So this is kind of showing the pathway, and I have to take a step back because one thing that I should have talked about in the last class but didn't was something interesting about protein kinases. And that's that they have this what's called an activation loop. And for most of them, I don't know if it's all, but it's definitely most, this activation loop has to be phosphorylated for the kinase to have full activity. And in the absence of that, they have very weak kinase activity. And that becomes important for how these receptor tyrosine kinases work. And I'm recording this afternoon lecture instead of doing the morning one because I messed something up quite badly. And I went back and looked up some more recent papers, and now I've got it all sorted out. So bear with me. So initially, this is a receptor tyrosine kinase, and we call these RTKs. Okay? And these kinases have very weak activity. So I like to think of them as like this kind of like weakly, they can't really, they're not phosphorylating anything. What happens is they bind to their ligand, and this example is EGF, okay? And what happens upon ligand binding is it allows them to dimerize. And what I messed up this morning was there's always been kind of two schools of thought. One was that the signal itself is a dimer, and then the receptors come together on that dimer of signal. And the other is that binding signal allows the receptors to dimerize, which is what they've shown here. So once they bind signals, it alters their structure so that these now dimerize. The end result is the same, but now this appears to be more in line with what people think, that it actually, the signal itself doesn't have to be a dimer. So that's what I messed up this morning. But the importance of this is that even with weak activity now, the receptor neighboring it is so close that even though it has this very weak activity, bam, it phosphorylates its neighbor on the activation loop. And now that neighbor's wide awake, so blam, it phosphorylates back, and then back and forth, and back and forth, and back and forth. So we call this trans-autophosphorylation. And we end up fully activating and creating a whole bunch of phosphorylated tyrosine residues in the cytosolic domain. Okay? Oh, I should point out one other thing. Sorry to jump back. One of the ways these were originally identified, and the first growth factor that was, or one of the first growth factors that was identified, was through a study of cell culture. And remember when we talked about cell culture, we talked about how we add serum, which is clotted blood plasma. Initially, some studies, people looked at just taking blood plasma and adding it to the media. And so plasma has had the white and red blood cells removed, but the platelets are still there and they're not coagulated. And this didn't allow this many cells to grow. Upon coagulating the platelets, which forms serum, it was found that serum could support growth. And it turns out that the platelets, when they coagulate, release a growth factor called PDGF, which is platelet-derived growth factor. This is a different growth factor. This one is epidermal growth factor, or EGF. Sorry to bounce back and forth there. This is a nice view, and it's showing us some of the importance of what happens here. But what we see is now we've created a whole host of binding sites for other protein binding partners, some of which may in turn become phosphorylated by the kinase activity, either turning them on or creating docking sites. Okay? So this allows things to dock to them. Here we can see the structure of two of these. These are VEGFR shown from two different angles. We can see the receptor and the signal. And here's a different one called KIT. Again, receptors and signal. Again, we're doing this little trick here where we're only looking at the extracytosolic domain because that transmembrane region is hard to crystallize and to determine the structure of. Okay, questions so far? So it's a really cool little system that binding the signal allows them to, in this case, dimerize. And even though they have that very weak activity, it's enough because you've brought them so close together that they will ultimately phosphorylate each other. Okay. We're going to talk about a protein called RAS now. And RAS is a monomeric GTPase. And there is something called the RAS superfamily. And the RAS superfamily is all the monomeric GTPases. Okay? There's about 100 of them in the human genome. Okay? And RAS is one of them. And it's the one the family is named after. This was originally isolated from a rat tumor called a sarcoma, and so it was called RAS. In fact, RAS is implicated in something like 30 to 50% of all human cancers. It's something called an oncogene which promotes growth. So just a little note about nomenclature. When genes were named, they were often named from either where they were isolated or what the effect was. And again, we often didn't know exactly what was happening at the DNA level because discovery of a lot of these things predated our ability to efficiently sequence them. And so sometimes we'll see that a gene has two names because it was discovered because it was implicated in two different pathways or two parts of a pathway. And then later people determined it's the same one. And you've probably come across that before, I would think, when you've been studying. So there's there's a number of rules that people use for naming genes. And there's like a renegade group of geneticists. And these were the early Drosophila people. And they've kind of toned this down now. But Drosophila was famous for having somewhat more fun, sometimes objectionable names for their genes. And a lot of these have been renamed, but the originals, they've kind of held on to it. And so we see a couple of sort of fun examples here. The first one is son of sevenless. And so Drosophila, the genetics are very well understood, but we also understand the development very well. And so it's a model organism for studying development. And we know what each cell is going to divide and become. So it's been mapped, so they know what cells are going to become. And researchers found that if they knocked out the gene called sevenless, what happened was a very important cell called R7 in the development of the eye, so the letter R7, didn't form. And so they called this mutation sevenless, okay? Now, downstream of sevenless was another protein involved in the pathway, so they called it son of sevenless, okay? And so that's Drosophila genetics for you. So RAS is up here bound in the membrane. A monomeric GTPA is in its off state, GDP bound. Its GAF is son of sevenless. But son of sevenless is cytosolic and floating around, and we remember that the cell is chock full of macromolecules and molecules and all kinds of things, and so it doesn't efficiently get to RAS. Son of sevenless is also bound to GRAB2. GRAB2 has a domain that binds to a phosphorylated tyrosine residue. So when we activate the receptor tyrosine kinase, we bring GRAB2 to the membrane. And this allows son of sevenless to activate RAS, the monomeric GTPAs. And we'll look at the important effect that has in a couple of slides. Any questions? Yes?
I was wondering if you could repeat the situation for the eye development where one of the
Oh, yeah, so sevenless is missing a cell called R7. So it's actually one of the differentiated cells of the eye doesn't develop properly. And so a lot of these Drosophila mutations, they're developmental mutations because that's what a lot of the studies surround. Yeah. And we'll see that in the next couple of slides. So we put together, I just thought it would be fun to share some of them with you because some of them are kind of amusing. So there's one called LUSH, and this gene is associated with attraction to ethanol. There's one called Kenny that dies after 2 days, like Kenny from South Park. There's one called Ken and Barbie, a gene which lacks external genitalia, as do Ken and Barbie. There's one called Tin Man, which has no heart. There's one called Kojak, which Kojak was a famous 70s detective that was a bald guy that always had lollipops instead of smoking because it was supposed to be a positive imagery, I guess. And so Kojak doesn't have the little sensory hairs on the Drosophila. And there's lots of other ones out there. And we'll look at a couple of them in next class too. But anyway, that's just a diversion. So let's look at what happens here, because this turns out to be pretty cool. So RAS is part of what's called the MAP kinase pathway. And MAP kinase stands for mitogen activated protein. And the mitogen activated proteins are transcription factors. And in this example here, we have ETS and JUN are the two. These are the two transcription factors, which are mitogen activated proteins. A mitogen is a little bit different from a growth signal in that it's there to initiate replication. So it starts the cell cycle off. So you have these two factors or families of factors are all working together, but we can actually separate them, but we're not going to do that in this class. We're not going to really concentrate on whether it's a mitogen or a growth factor. Okay? But that's what a mitogen kicks off the cell cycle. Now, the mitogen activated protein is not active unless it's phosphorylated. And so, of course, to phosphorylate it, you have a mitogen activated protein kinase. The mitogen activated protein kinase is not active unless it's been phosphorylated. So lo and behold, it's phosphorylated by a mitogen activated protein kinase. This example is looking at one called MEK. The mitogen activated protein kinase is not active unless it's phosphorylated, and you'll never guess what phosphorylates it. A mitogen activated protein kinase, which in this example is this protein here called RAF. These three kinases are physically associated on a scaffold. So it's an interesting example where these kinases don't amplify a signal because they pass it directly between them, but they can be separately regulated. The other important thing here is we're back to our old friend combinatorial. There's actually seven of both the MAP kinase and the kinases, and there's 12 of the MAP kinases. And so these can be put together in different combinations to influence different pathways. So kind of a neat system. And again, we see our old friend combinatorial rearing its head. Okay? Questions about? Yes?
So do they only interact with each other?
Not necessarily. So they can still be influenced by regulatory proteins, is my understanding. But due to proximity, they're most likely to just interact with each other. They're physically associated with each other on what's called a scaffold. And a scaffold is just a protein that holds other proteins together. Yeah. And we don't go hugely into the detail of that either here, but you will explore that more in Biology 360 if you take it. So I've switched the kind of order of these two slides, the next two slides, because here we are back at our friend insulin signaling pathway. And this is the slide I showed you the other day. And one of the things that you'll see is that, oh, lo and behold, it can recruit son of 7-less. Okay? And so this can initiate the MAP kinase pathway. Another important pathway that's mediated by insulin is, this is called the PI3 kinase pathway. So this is lipid signaling. So here is our friend, phosphatidylinositol 4,5-bisphosphate, and it's sitting in the membrane. It can be phosphorylated at position 3 by an enzyme called PI3 kinase. PI3 kinase is recruited to the membrane where it can phosphorylate, PIP2, when it binds to the tyrosine kinase receptor indirectly. So what happens is IRS1, this one, binds to the receptor tyrosine kinase. It gets phosphorylated. And then PI3 kinase binds, which brings it to the surface or the membrane, which then will phosphorylate it, okay, to produce PIP3 which is phosphatidylinositol 3,4,5 trisphosphate. Don't worry about that. This PIP3 binds to a domain called a PD domain. And so now a whole bunch of proteins that have PD domains get recruited to the surface, or sorry, to the membrane. They get recruited to the membrane. This leads to activation of a protein called AKT. And what AKT does is it will activate a glucose transporter called GLUT4 that brings in glucose from the blood. Because remember, insulin signaling is in response to elevated blood glucose levels. So the opposite of glucagon. So there's too much glucose in the blood, the liver activates this pathway, and now glucose is brought in. The other thing that AKT activates is glycogen synthase. So what this does then is this brings the glucose in, and it can be constructed back into glycogen for storage. So that was that balance I was sort of referring to a little bit earlier. The whole pathway is shown here, but what I want to show you this for is there's something really interesting here. So first of all, remember when we're looking at these pathways we follow, and the arrows are positive activators and the little like T-bars are negative activators. So like this PRAS40 inhibits mTORC1, for example. So AKT, we can see, activates two proteins. And look at this. This is what's so cool. So that glucose transporter is present in a vesicle. And when we activate the pathway, it causes the vesicle to fuse with the plasma membrane. So here it's not so important for releasing the contents of the vesicle, but rather, remember that vesicular membrane now is fused with and becomes part of the plasma membrane. So now the glucose transporters are embedded in the membrane where they can bring glucose in. Once glucose levels have been rebalanced, this will go back through an endocytotic process to being in a vesicle. Okay? So pretty cool stuff going on there. We'll look at a couple other aspects of this a little bit later. So I always like to point out that grumpy old people like me, well, not actually me, but grumpy old people of my generation often complain about how, oh, everything's so easy for kids today, and they all complain, which we know is actually kind of a bunch of garbage, but things are just different. And here's a good example of that. So this is the pathway that people today have to learn. This is taken out of my third-year textbook, and it's the exact same pathway, and this is all that was known about it. So like studying the signaling pathway when I was going through school, that was like the easiest section because it was all like physically there would be question marks all over the place and dotted lines. So anyway, I'm not going to make you memorize it at this level of detail, but it's pretty neat to see just how far we've come in these signaling pathways. And this is just looking at insulin signaling. Remember, we have all these other pathways, all these other receptors, and their pathways. So that's as far as we got this morning. I hope everyone has a great day. I hope we see some of you tonight at our social hour. And otherwise, we'll catch up with you on Friday.